Hello students. Students, in my previous video we discussed about the transcription unit, and now today we will discuss about the whole process of transcription. As I told you earlier that the transcription is the process of copying the genetic information from one strand of DNA into messenger RNA. This transcription actually consists of three important steps, and we will see what are those three important steps. So the three important steps are: first is initiation, second is elongation, and the third one is termination. So the three important steps of transcription are initiation, elongation, and termination. Here we can see a DNA helix. This DNA helix has two strands, a template strand having the polarity 3' to 5' and a coding strand having polarity 5' to 3'. In between the two strands there is the hydrogen bonds present. So what happens when the process is to be initiated or when the process of transcription is to be started? The RNA polymerase, it will bind with the sigma factor. The sigma factor will come and it will bind to the RNA polymerase. This sigma factor, as I told you earlier, it helps us to recognize the promoter site. Second important thing, the process of transcription will be initiated only and only when the sigma factor will bind to the RNA polymerase. So, in this promoter site, what will happen? The RNA polymerase will bind and now it will break the hydrogen bond. The second step is elongation. During this process, what happens? When this RNA polymerase moves, it will break the hydrogen bonds and the ribonucleotides. Remember, ribonucleotides, because we have to synthesize RNA. So ribonucleotides, which are present in the form of triphosphate, they will come and they will lie opposite to the template strand. This is our template strand, which has been shown with the red color. So what will happen? The ribonucleotides, they will come and they will lie opposite to the template strand or antisense strand. I told you it is present in the form of triphosphate, that is ATP, GTP, CTP and UTP. Clear? Now this RNA polymerase, what it will do? It will form the phosphodiester bond between them. So during elongation, RNA polymerase, it move along the template. Second thing, the ribonucleotides, they will come and they will lie opposite to the template strand and a phosphodiester bond will be formed between them. My dear student, one thing you have to remember, this process of transcription requires energy, and how does this energy will be met during this process? For that, an enzyme, remember, pyrophosphatase. This enzyme hydrolyzes, it will hydrolyze 2-phosphate from triphosphate, and when it will hydrolyze 2-phosphate, what will happen? Energy will be released and that energy will be used for the process of transcription. The third step is termination. RNA polymerase will move and it will reach to the terminator site. Terminator site is that site where the process of transcription will stop. And to stop it, it requires a factor, and that is our Rho factor. In case of prokaryotes, Rho factor is required for the process of termination, and this rho factor is actually a type of specific protein. Now what will happen? At the terminator site, the RNA polymerase will be removed, the rho factor will also detach, and we will get the RNA. One thing you have to remember, the whole process of transcription in the case of prokaryotes, it is taking place in the cytosol. But in eukaryotes, it takes place inside the nucleus. One more important thing, the RNA which is being formed, or the transcription process is taking place in the cytosol, and in the same time the process of translation also starts in the case of prokaryotes. Clear? So this was all about transcription in prokaryotes, and now we will discuss the process of transcription in eukaryotes. Students, now we will discuss transcription in eukaryotes. In eukaryotes, the process of transcription takes place in the nucleus, while in prokaryotes, it takes place in the cytosol. Now, after the process of termination in eukaryotes, we got the hnRNA. hnRNA is heterogeneous nuclear RNA. This hnRNA has certain coding and non-coding sequences. The coding sequences are known as exons and the non-coding sequences are known as introns. So to get the messenger RNA, there is processing of hnRNA. Now when we do the processing of hnRNA, it includes various steps, that is capping, tailing, splicing, and ligation. So let's see what are these capping, tailing, splicing and ligation is. So as I told you that we got the hnRNA. This hnRNA is very unstable. So what we do is, at the 5' end of this hnRNA, we add a cap-like structure which is made up of methyl guanosine triphosphate. Do remember, at the 5' end, we add methyl guanosine triphosphate and it forms a cap-like structure. Similarly, at the 3' end, we add 200 to 300 nucleotides of adenylate acid. And it is known as poly A tail. So what we have done? We have done capping and tailing. Capping is done at the 5' end, where methyl guanosine triphosphate is added, and at the 3' end we add 200 to 300 nucleotides of adenylate acid, which is known as poly A tail. After that, what we do? That the introns is removed with the help of an enzyme called spliceosome. Now what is this spliceosome? Just see here, there are certain proteins required for this, and these certain proteins are called as SNRPs. SNRP stands for small nuclear ribonucleoprotein. This protein, along with some other protein, forms a complex, and that complex is known as spliceosome. So what does spliceosome do? It do the splicing. It cut and it removes the non-coding sequence, that is introns. And after that, what happens? We have only the coding sequences, and these are attached with the help of the enzyme ligase. So that is ligation. After that, we will get our messenger RNA. So the process of transcription is over here. The messenger RNA is synthesized, and this messenger RNA will come out of the nucleus and it will go to the cytoplasm. So this was all about the process of transcription.